Next up for us is genetics. Obviously, in just a first look, it's clear we're not going to cover this in detail. I just want to touch on a few things. Here's what I think of genetics. I wasn't a straight A student in college, but I got more A's than B's and more B's than C's. The only D I ever got was in genetics, and actually it was a D-. Obviously, genetics is amazingly complex, and look up things like epigenetics to see that there's still a whole lot we don't understand at all. Think about it this way, too. Cancer kills a lot of us. Cancer is a genetic issue where cells replicate when they shouldn't. While there's a lot of opportunity to think at different levels and gain mastering skills in genetics, my D-, tells you I'm not the one to do it. But definitely look into it. We're going to talk about basic genetics. We'll talk about modes of inheritance, mutations, and then we'll cover some major genetic crosses. So genetics is the study of the mechanisms of heredity. Ella has soccer practice, evidently. How long has that been there? In the last 10 years, we sequenced all the human genome, so we know the basic code. That has caused a burst in our understanding. Quick story, and I want to cover the basics of genetics. When my wife was pregnant with our second child, we got some genetic results back that indicated that my wife is a carrier for cystic fibrosis. It's one of those things where they ask, do you want these testing? You sign it, and you even forget that you've even had it until you get this letter. And so we got this letter that said, your wife's a carrier for cystic fibrosis. Because I had some genetic training, I knew I needed to get tested. And ironically, this is 3 years after I'd actually done research on cystic fibrosis, but had left to go into education. So I came back as not being a carrier when I got that test. And with that knowledge, though, I could do the Punnett square in my head quick and realize our child in the womb would not have cystic fibrosis. The reason you need to be careful about this is because cystic fibrosis, usually what kills a person with cystic fibrosis is a bacterial infection. And so if your child does have cystic fibrosis, you want to know from day one so that you can protect them from these infections. So I knew there was a 50% chance my daughter would be a carrier. In fact, my daughters still have a 50% chance of being a carrier and a 50% chance that both of them don't have the mutation at all. So we haven't had our daughters tested yet, but I know that if they have children with another carrier, there's a 25% chance that my grandchild could have CF, a 50% chance that the child is a carrier, and a 25% chance that the child does not have the mutation. I believe that there's a 1 in 20 chance, something like that, that you're a carrier for cystic fibrosis. So this is not an insignificant chance that my kids will procreate with another carrier of cystic fibrosis. So some of us already have training in basic crosses of dominance, recessive, and X-linked disorders. But for those that have never heard of a Punnett square, I think we should cover some basic genetics in AP2. We won't do dihybrid crosses as if we're trying to find unique flowers and things like that. We're not trying to be Mendel here. But you should know for each of the 3 types of crosses, dominant, recessive, and X-linked, what are the genotypic and phenotypic ratios that the offspring would be or could be. First, note that genetics is obviously derived from the word gene. A gene is made of DNA. We have a pair each of 23 DNA molecules called chromosomes. A chromosome has a number of uncoded areas, but also areas that have genes. A gene code breaks down into triplets, 3 bases of adenine, guanine, cytosine, and thymine. So those are the A, T, C's, and G's. The 3-letter sequence of these base codes for an amino acid. A sequence of amino acids is a protein. So essentially, a gene is the instruction for how to make a protein. So DNA is instructions for how to make a polypeptide or protein. So when we think of genes, we're thinking of proteins, whether these proteins code for eye color, hair color, that's my daughter Celie, or even the type of blood you have. So we're primarily a collection of proteins, obviously. So then mutated genes cause changes in proteins. In muscular dystrophy, a protein that connects muscle to tendons is weak. So when the muscle pulls, it doesn't always pull the bone very well. And you can actually see that the muscle has shrunk away from the tendon in muscular dystrophy versus normal. In cystic fibrosis, a chloride channel is deficient so that chloride can't be transported across the cell membrane. We've talked about this before, that you want to move chloride so the body can move sodium, and by moving sodium chloride, the body can move water. And by moving water, the body can dilute and clean up mucus. So without this channel, mucus builds up in various body areas, tubules of the reproductive system, the pancreas, and the lungs. And achondroplasia, there's a mutation in the off switch for a molecule that signals to stop growing. With no off switch, the molecule continues to send the signal to stop growing, and so growth is inhibited. Another example would be PKU. The enzyme necessary to break down phenylalanine doesn't work, and when phenylalanine builds up, it damages neurons. Lastly, people who survived the Black Plague did so because they lacked the receptor necessary for the pathogen to enter cells. Since HIV uses the same receptor, descendants of survivors of the Black Plague are immune to HIV. I don't want to talk on this much, but proteins can be mutated in many ways. So while we talk about cystic fibrosis as a single disease, there are many different ways that the protein can fail, and all due to different genetic mutations. So these are some of the common mutations that cause cystic fibrosis down here. Maybe the protein isn't made. So here's the normal cystic fibrosis. It's called the cystic fibrosis transmembrane conductance regulator, the CFTR channel. So here's normal. Maybe the protein isn't made. Maybe it's made, but it's destroyed too early, so there's a block in processing. That's the most common one, is delta F508. And that one's kind of interesting because it just means there's not a phenylalanine, amino acid phenylalanine, at the 508th amino acid position. Maybe there's a block in regulation, so the channel is there, but you can't turn it on. Maybe the channel is there, but it doesn't allow chloride to go through. Or maybe there's just not enough of it made at all. So maybe the protein isn't made. Maybe it's made, but it's destroyed too early. Maybe the protein is made and would transport chloride, but the channel can't be turned on. Or maybe the channel doesn't let the normal amount of chloride through. So lastly, maybe the protein is made correctly, but not enough is made. There are two interesting things about the delta F508. First, any amino acid at the 508th position besides phenylalanine will cause a mutation. So it has to be a phenylalanine. Also, the protein actually works, but when the Golgi apparatus sees the change at 508, it decides the protein is made wrong and sends it to the lysosome. So a strategy to cure many people with this particular mutation is to trick the Golgi to let the protein get to the cell membrane where it will actually work. One last interesting thing is that it shows how curing a disease can be so complex because different mutations lead to different diseases, and this may require a lot of different mechanisms to cure the disease. So potentially there's not one cure for all of cystic fibrosis. You might have to cure each of the problems individually, which sure makes things a whole lot more complex. Have I mentioned yet that genetics is pretty complex? A nice picture of your 23 pairs of chromosomes. Well, not yours, but human chromosomes. Notice how X and Y are different. So X and Y are a little bit different down here. Females have two X chromosomes. Males have a Y. So these would be the chromosomes of a male person. You have two copies of every gene. So each copy is called an allele. So two copies of every gene. An individual gene is called an allele. Eye color is more complex than this. There are things like hazel eyes and sunflower eyes. My daughter Celia has sunflower eyes where they're brown in the middle and blue on the outside. There's also heterochromia. JJ, my wife, has heterochromia. That's where part of one of her eyes is blue and part of one of her eyes is brown. Or you can have one eye color that's blue and the other eye is brown. That's also heterochromia. So it can be much more complex than we're going to deal with it, but let's pretend it's simple. In a simple sense, you can have a brown eye allele, a blue eye allele, or a green eye allele. If both alleles are the same, then it's called homozygous. If the alleles are different, it's called heterozygous. When you only need one copy of an allele to see its effect, the allele is called dominant. So if one allele for eyes is brown, it doesn't matter what the other allele is, the eyes will be brown. If you need both copies to be homozygous to see the effect, then the allele is recessive. Another way to think about this that often works with mutation is that if a mutation is toxic, it will be dominant. For example, in Huntington's, long repeats in the DNA lead to long proteins that build up in the brain. This protein is toxic, and so Huntington's is dominant. If one copy of a protein is enough to do what you need to do, then the allele is recessive. For example, cystic fibrosis is a mutation of a chloride channel. As long as one allele makes the proper proteins and can make the chloride channel, the disease is not evident. But if both alleles make a faulty protein, that's when you see the disease. So cystic fibrosis is recessive. The slide is just to practice your terminology a bit. The parents are heterozygous and have brown eyes. The child on the left is homozygous dominant with brown eyes. The next two children are heterozygous with brown eyes. And the last child is homozygous recessive and has blue eyes. Genotype refers to the identity of the alleles, so saying that someone is homozygous dominant is stating the genotype. The phenotype is how the trait is revealed, so brown eyes versus blue eyes. So the genotype here is big B, or big B, little b, and the phenotype would be brown eyes and brown eyes. When it comes to alleles, it's best to mix them up to create genetic diversity. Differences in genetics will protect a species. For example, again, the survivors of the Black Plague contained a different receptor than most of the population, and that receptor did not allow the pathogen to infect those people. So survivors of the Black Plague are also immune to HIV since HIV uses the same receptor. So even if HIV were more potent than it is, which is scary to think about, it still would not kill off the entire human population because some people have a genetic difference that protects them from this virus. There are 3 ways to increase genetic diversity: crossing over prior to meiosis, independent assortment, during meiosis, and random fertilization. Other examples of why genetic diversity is good is that differences in elm trees allowed this type of tree to survive, because while the American elm was decimated by Dutch elm disease, not all elm trees were destroyed. A cautionary tale is that all cheetahs are very genetically similar, and so they're more susceptible to disease and genetic mutation because of their lack of diversity. Hey, there's a quick goal there. Soccer. In meiosis, and after the cell has reached 92 chromosomes, it must divide twice to get down to 23 chromosomes. So we talked about this when we did genetics, that you go up to 92 so you can mix up the genes a little bit, but then you have to divide down to 23 chromosomes in sperm, and 23 chromosomes in egg, so when sperm meets egg, you're back up to 46. When you go through these divisions, chromosomes don't stick together. Each pair of chromosome will divide up into the resulting cells independently. So if, say, chromosome 1 and 2 always stuck together, the diversity would be reduced. But since each chromosome acts on its own, this increases genetic diversity, and this is called independent assortment during meiosis. A metaphor would be that if you have 23 bags, and each bag has both chromosome 1s, and the next bag has chromosome 2s, and so on up to bag 23, the chromosome that goes into one or the other cell is completely random. So by keeping things independent, there are 8.5 million different combinations that will ultimately go into a single sperm or egg. We've talked about crossing over when we covered meiosis as well. In meiosis, the chromosome number doubles from 46 to 92, so that homologous chromosomes can exchange genes. If this did not occur, variability would be limited in that some genes would always stay together, so blonde-haired people would always have blue eyes and brown-haired people would always have brown eyes. But crossing over allows the genes to mix that up a little. So an example, again, sorry to be redundant, an example of the result of crossing over is that the genes for hair and eye color are on the same chromosome. So there's certain combinations that are more likely to occur, like brown hair and brown eyes, blonde hair and blue eyes, red hair and green eyes. But because chromosomes sometimes cross over, randomness is introduced, and there's some brown-haired individuals with blue eyes and blonde-haired people with brown eyes. Some examples: Google blonde hair and brown eyes, and there's something sort of weird about that one. I couldn't find any that were copyright free, so I couldn't put them in this public space. But Google blonde hair and brown eyes. That's almost more weird than brunettes with blue eyes. The last thing that increases randomness is that many more sperm and eggs are produced than are needed. So we talked when we did reproduction that there are 7 million oocytes originally made in females, and many, many more sperm than that. So since only one egg and one sperm results in fertilization, an offspring represents 1 out of 72 trillion possible zygote combinations. Each of us is a 1 in 72 trillion. I feel special. That's basic genetics. We'll next go into modes of inheritance. So how do we sort this all out when it comes to what are my kids going to have genetically? Well, we'll keep it pretty simple here, but the easiest tool is a Punnett square. Because of independent assortment, a child may receive either of the parents' 2 alleles for a gene. So the Punnett square puts the 2 possible alleles a child can receive from one parent along the top of the box, and the 2 possible alleles a child can receive from the other parent along the left side of the box. In this particular Punnett square, paternal father is put on top, maternal is put on the side, mother is on the side, but it could be the other way around too. Could put the mother on the top of the square and the father on the side. Then you can kind of sort out that the offspring can receive any one of those 4 combinations of alleles. In this case, if the parents are heterozygous, we can see that the offspring can receive 2 big B's, a big B and a little b, another big B and a little b, or 2 little b's. So we can see that the offspring can be big B, big B, big B, little b, big B, little b, or little b, little b. If this was eye color, the parents have brown eyes because they're heterozygotes. 3 of the kids have brown eyes, and 1 kid has blue eyes. So you can see that there's a 25% chance that these parents will have a blue-eyed kid. I don't want to cause any stir, But if your parents have brown eyes, you can have blue eyes. But if your parents have blue eyes, it's not possible for you to have brown eyes. I did have a student question that once, and when she went home, her father told her that he was in a welding accident and that had bleached his eyes. I'm not really sure about that story, but I'm pretty sure you can't have brown eyes if your parents have blue eyes. Maybe you do the genetic cross yourself to see that, because if you've got blue eyes takes little b, blue eyes takes little b, there's no way to get a big B if the parents have two little b's. We will soon go over some of the more complex genetic mechanisms of inheritance, but the three main types of inheritance are autosomal dominant, autosomal recessive, and X-linked, also called sex-linked because it has to do with the sex chromosomes, or the chromosomes that determine male and female. Autosomal refers to the non-sex chromosomes, so chromosome 1 through 22. The sex-linked chromosomes, chromosome 23, has different genetics because the X chromosome is much bigger than the Y chromosome, and it has many more genes. The basic reason X-linked is different is that in females with two X genes, a mutation generally follows recessive genetics because while one X might have a mutation, there's another X chromosome that may make the protein properly. But with males, if there's a mutation on the X chromosome, the Y doesn't have the genes to act as a backup. So for the sex chromosomes, diseases have different frequencies in females versus males. So for example, males are much more likely to have hemophilia. Males are much more likely to be colorblind because those are mutations on the X chromosome. We touched on this earlier when just defining dominant versus recessive. A dominant mutation will be expressed if only one mutant allele is present. So if this were Huntington's and there was one mutation, let's say the whites, if only one copy is there, then the person is affected. So just one copy of the white is enough to be affected. Dominant mutations are usually relatively rare as they're selected against. If the mutation is significant enough, it prevents reproduction. A classic example of a dominant mutation is Huntington's disease, where if you have one mutant allele, you will develop Huntington's disease. The problem with Huntington's disease being selected out or not being selected out is it doesn't show or rear its ugly head until you're past reproductive age. Now that we have testing and people can be tested to see if they have Huntington's, they might make reproductive choices that will eventually lead to Huntington's going away. I don't know that people always make those decisions. It was a factor that factored into us having a third child, knowing that my wife is a carrier for cystic fibrosis. Maybe you don't want to pass that on to another child, and so let's just stay with two kids. With recessive, both alleles must be mutated to see the disease. An example is cystic fibrosis, where a chloride channel is mutated. If only one allele is mutated, the other allele will make enough functioning chloride channels so no disease is evident. But if both alleles are mutated, no functioning chloride channels will be made, and so cystic fibrosis is present. So here again on the image, you need two copies to be affected. Or in the case of cystic fibrosis, white means a chloride channel that doesn't work. You need two channels that don't work. As long as you have one channel that works, you're not going to have cystic fibrosis. So here's a nice picture of the X chromosome and the Y chromosome. You can see how much smaller the Y chromosome is. About the only gene on the Y chromosome is the gene to grow hair out of your ear. So if a female has a mutated X, the other X might still make the correct protein, and so genetics are similar to recessive genetics. If a male has a mutated X, so over here the male has a mutated X, the Y can't recover it for him, or the Y is no backup, and so the genetics is more like dominant genetics and the disease will be evident. Classic examples are hemophilia or color blindness. Because females have a backup again, they're less likely to get X-linked disease, and things like hemophilia and color blindness are much more common in males. So in this case, the female has one bad X gene, one good X gene. The good X gene makes the blood clotting factors or makes the cone necessary to see all the colors, and it makes up for a mutated X chromosome. Remember rods and cones in the eye? That's why I said cone. Rods see black and white, and you have different cones to see color. So I gave some classic examples of each type of inheritance, but here are a few more if you'd like to look them up. Achondroplasia is a condition where a molecule that is to turn on and turn off a growth factor is mutated to keep sending the signal to turn off the growth factor. It's kind of hard to follow, but it's a failure of the off switch on the off switch, and so essentially growth is always turned off. So even if one allele is not saying turn off growth, the fact that one allele is saying turn off growth means growth is reduced. So dwarfism is dominant, or at least this particular case of dwarfism. You may wish to research the molecular mechanisms of more diseases to hone your mastery thinking. A new term here is that if, for recessive crosses, a person has one good allele and one mutant allele, the person is called a carrier. They carry the mutation, but they do not show it. So my wife is a carrier for cystic fibrosis. She has the genetic mutation. She can carry it on to her kids, even though it doesn't cause the disease in her. There are times when the carrier is different than the normal and the affected. So the normal might have no effect. The homozygous recessive has significant disease, and the carrier has some disease. This is called incomplete dominance because the heterozygous individual isn't like the dominant or the recessive person. Sickle cell anemia is a classic example where both carriers and homozygous recessive both show signs of the disease. If you have both mutations, you have much more significant disease than if you only have one mutation. So here the small s indicates the mutation. Multiple allele inheritance. In most cases, they're not just two possible alleles, they're multiple alleles. So there's multiple genes for, in the case of eye color, it would be brown, blue, and green. There's multiple alleles. Another classic example is blood type, where there's an A allele, a B allele, and then a null allele or a nothing allele. So recall that from blood typing, that blood type is caused by proteins on the outside of red blood cells, and the nothing allele results in no protein being made on the red blood cells, so that would be O type. A and B are codominants, and no allele is technically recessive. So if you have the A allele, you have A-type blood. If you have the B allele, you have B-type blood. You can have both A and B alleles, which would give you AB-type blood. And if you have neither alleles, that's when you have O-type blood. So A and B are codominant. We've kept things pretty simple and pretended that things like eye color are caused by just one eye color allele. Most phenotypes are due to a combination of different genes. So eye color, skin color, height are due to multiple genes and multiple alleles, and this is called polygenic inheritance. All right, so I kind of made a mistake on this figure because this says polygenic inheritance of skin color, which is what this figure used to be. But when I moved to copyright free, I had to find a height one. So it works the same for skin color and height. Essentially, there's multiple genes, and the combination of those genes create a continuum of height or skin color. So two people with a particular skin color can have a child with a particularly different skin color. There are multiple examples that demonstrate that genetics is much more complex than what we understand. A classic one was that it was found that there were two different diseases with the same genetic mutation. The DNA looked the same in both conditions. There was a deletion in chromosome 15. It turned out if the mutation came from the dad, the result was Prader-Willi syndrome, a condition with obesity, MR, and short stature. If the mutation came from the mother, the result was Angelman syndrome, a condition with laughter, jerky movements, and motor and mental deficits. The genetic anomaly pointed out that it isn't just the sequence of DNA that determines phenotype. There must be more. The more looks like it has to do with methyl chemical groups that are added to the DNA to turn on and turn off genes. So the Prader-Willi syndrome and Angelman syndrome was replicated in mice, an animal with well-worked out genetics. So the seeming identical genetic mutation leading to the two different phenotypes appeared authentic and not just a result of the complexity in human behavior. What it came down to is something called epigenetics. So the study of how these methyl tags might turn genes on and off is called epigenetics. If you'd like to hear more about this, watch Ghost in Your Genes from Nova. You can find copies of the movie on YouTube. Interesting, or at least I find it interesting, that two different versions of the movie were made for the US and British markets. The US market movie focused more on the connection between epigenetics and cancer, whereas the BBC version took time to explore how the fear that arose from 9/11 may have caused epigenetic changes that will play out for generations in American kids. The bottom line for epigenetics is that the sequence of DNA changes slowly, but the methyl tags might change more rapidly, and they may change due to human behavior. This means that you're changing your genes with your behavior. If this is true from a very young age, you're already a parent to your kids because you're changing the DNA you will pass on to them. Watch Ghost in Your Genes because it will go into a whole lot more detail. I do have an infographic on this if you're interested. You should be able to zoom in on this, at least on the Prezi, and see more of the detail. So these are some things that we know affect epigenetics. So red wine, BPA, most of us have heard to avoid BPA. how you're nurtured. Nutrition seems to play a role. Things that we know of how epigenetics affects behavior. So we know that we can clone cats and they don't look the same, even though they're identical genetically. The Human Genome Project has pointed out that humans and chimpanzees are very similar genetically, but there has to be something really different about the DNA in order for us to have this different, obviously, this different animal. So we've known for a long time a little bit about epigenetics and methylation because we know that every cell in your body has the same DNA code. But sometimes that DNA code leads to you making a cell that's going to make the liver, or it might lead to making a neuron. So clearly in each cell, you turn on your genes in different ways. And then, of course, Angelman syndrome versus Prader-Willi. Some of the things that epigenetics might affect, it means that you're a grandparent already. Some of the best examples that led to this was Marcus Pembry had this data that showed how much food grandpa had was related to the likelihood of type 2 diabetes that the grandson had years later. And so it seems that whatever the grandpa does can affect the grandkid. Anyway, look more into epigenetics because I think it's probably going to play an increasing role in our understanding of things like cancer and pesticides and things like BPA, and how you raise your kids might affect their behavior for many, many generations, or the behavior of your grandkids and great-grandkids. Mitochondria also has their own DNA. A problem with the mitochondria is that they don't have the quality control and replication that DNA in the nucleus has, so there may be more mutations in mitochondria. Interestingly, all mitochondria DNA is passed on from the mom, so kind of a cute little name to call it is Eve DNA, as if all your mitochondria was passed down to you from Eve because it's always been passed down from your mom. Newborn infants are screened for a number of genetic disorders: congenital hip dysplasia, imperforate anus, PKU. Genetic screening alerts new parents that treatment may be necessary for the well-being of their infant. Another example, obviously, is our screening for cystic fibrosis. Identification of the heterozygous state for a given trait. So carrier recognition is finding the heterozygous state. So two major avenues are used to identify carriers: pedigrees. Pedigrees trace a particular genetic trait through several generations, and it helps to predict the future. So it's going to predict the likelihood that the grandchild is going to have this genetic mutation. We can do that with things like cystic fibrosis. Blood tests and DNA probes can detect the presence of unexpressed recessive genes. Sickling, Tay-Sachs, cystic fibrosis genes can be identified by these types of tests. Pedigree analysis calculates the odds that an offspring will have a disease based on the family tree. So with diseases like cystic fibrosis, it's important to know if a child could have CF from the moment they're born so the child can be protected from infections that will shorten the life of their lungs and of them. You can kind of see that based on the different types of genetic cross, the pedigree looks different. And so you can then predict what type of disease or what are the possible outcomes of those crosses. Pedigree analysis is completely safe, but it only will give likelihoods. It won't tell you if this child has cystic fibrosis. So to be sure, a fetus can be directly genetically tested. Amniocentesis is when amniotic fluid is withdrawn after the 14th week, and sloughed fetal cells are examined for genetic abnormalities. Chorionic villi sampling is when the chorionic villi are sampled and karyotyped for genetic abnormalities. For even more techniques, simply search Wikipedia for fetal testing. So genetic testing now, we've gotten more advanced. Chromosome studies are, as we mentioned, pedigree and karyotyping. DNA studies, there's both indirect and direct, involve sequencing the gene or part of the gene, as in CF testing. Protein truncation studies are used to see if there's a change in the length of a protein, as some mutations result in a protein being shortened. Biochemical testing can see if there's a physiological sign of disease, like high phenylalanine, and the blood would indicate PKU. So those are modes of inheritance. I won't spend a whole lot of time on this, but I want to give some examples of mutations. A mutation is a change in a gene or chromosome that results in an abnormal trait. There's probably infinite possible mutagens, but some of the most common ones are cigarette smoke, pesticides, X-rays or radiation in general, ultraviolet light, and nuclear radiation. Three main types of mutation. Loss of function would include Tay-Sachs, PKU, and Niemann-Pick's. I'll give you one example, but maybe look up the other two. In Tay-Sachs, lysosomes are not as able to clear a molecule called a ganglioside. The gangliosides build up and kill cells. Partial function, like cystic fibrosis and sickle cell. In sickle cell, hemoglobin is not able to carry as much oxygen, and in low oxygen conditions, it makes cells that turn into a sickle shape. Hemoglobin actually kind of binds together into these long sticks, and those long stick-like structures push out the red blood cell into a sickle shape. So you're losing function of the red blood cell. Gain of function, and that may not be a good thing. Sounds like if you're gaining something, that's good. But if what you're doing is you're making more excitable neurotransmitter, then you may have too excitable of neurons, and that could lead to epilepsy. So there's many types of epilepsy, but there's a subset where ion channels are too excitable, leading to the electrical storms that characterize seizures. Aneuploidy. Whereas mutations are a change in the DNA sequence, aneuploidy occurs when there's an error in meiosis and a cell receives too many chromosomes. So remember, you're supposed to climb up to 92 chromosomes or replicate up to 92 chromosomes. Then you go through meiosis 1 and meiosis 2 to split back down to 23. Well, if there's a mistake in that split, you might end up with too many chromosomes in one of the cells. So aneuploidies are also referred to as trisomies because they're 3 chromosomes. And there's several examples. We'll go through some briefly. Classic trisomy is 21, Down syndrome. The risk of trisomy errors increase with the age of the mother. Classic signs of Down syndrome: large tongue, flat face, slanted eyes, slanted upward on the outside, single crease across the palm, shown here. It's interesting, too, that their pinky finger often is short and curves in. It's not really shown in this figure. But it's interesting how this one little extra bit of this tiny chromosome, so it's a relatively small chromosome, but too much of that DNA has a huge number of effects. Mental retardation is often present, but not always. So what I mean by multiple effects, like the tongue is usually large, the pinky finger is more curved, you have one crease across the palm, all because you have too much genetic information. Trisomy 18, classic signs are heart defects, displaced liver, low set ears, abnormal hands, severe retardation, and unfortunately 98% abort. The lifespan is usually less than a year. So to take off on that a little bit more, the reason to go through these trisomy disorders is to point out that too much DNA results in some pretty large genetic effects. It's interesting how too many plans for how to make DNA can result in such changes. So diverse of changes, so many diverse changes too. You can even see that as a trisomy occurs in larger chromosomes, the changes are more significant. So trisomy 21 has minor symptoms compared to trisomy 18, and trisomy 13 has much more significant effects than 18. So again, here's 21, which is pretty small. 18 is larger, 13 is even larger. So there's more effects the larger the trisomy. So too much genetic information is not necessarily a good thing. It's not a good thing, it's a bad thing. Classic sign of Patau, trisomy 13, cleft lip and palate, extra fingers and toes, that's polydactylism, defects in the heart, brain, kidneys, and most abort in less than a month. So Edwards syndrome, they often will last a year, but Patau rarely survive a month. Klinefelter is a trisomy of the sex chromosome with 2 X chromosomes and a Y. This one's interesting because then you have 3 sex chromosomes, which creates problems. It's not necessarily the 2 X's, because females normally have 2 X's. It's the 2 X's and a Y. You might ask, well, how do females deal with 2 X chromosomes then? Isn't that too much genetic information? For various areas of the body, what the female body will do, what the cells will do, is turn off one X chromosome and leave the other one turned on. That's why you get calico cats too. That's why almost all calico cats are female, because for that particular brown patch, one X chromosome is creating an X dye or protein pigment. In another area, the X chromosome is creating black, and so that's the only way you can get 3 colors on a cat like that, is to have 2 X chromosomes. Maybe look into that more if you're interested, because it's pretty interesting how that happens. So some females that have a mutation in sweat production, part of their bodies will sweat and part of their bodies will not sweat. So the body basically chooses, the female body basically chooses in large swaths which of the 2 X chromosomes to use. And so you're not using 2 X chromosomes in all the cells, and that's why you don't have too much genetic information in females. Classic signs of Klinefelter syndrome include breast development, small testes, sterile, low intelligence, not necessarily having mental retardation, but they clearly have the combination of female and male characteristics. One last one, Turner syndrome is a single X chromosome rather than two X chromosomes. So here, too little genetic information can create a problem too. So classic signs include short, absence of puberty, little estrogen, sterile, extra skin on the neck. Again, isn't that just so interesting that this one change in this relatively small chromosome creates a pretty big anatomical difference? So the top one is before surgery and the bottom one is after surgery. So it just seems so amazing that the genetics of this would result in such a dramatic change in how the body's structured. Those are mutations. Last up is crosses. So for our class, please be able to cross any combination of homozygotes and heterozygotes with dominant, recessive, and sex-linked mutations or X-linked mutations, and be able to give the phenotypic and genotypic ratios of the offspring. The genotypic ratio is the likelihood of each genotype. So for example, for two heterozygotes, the genotypic ratio is the likelihood of each genotype. So for example, for two heterozygotes, the genotypic ratio would be 1 big B, 2 big B, little b, and 1 little b, little b. The phenotypic ratio is the likelihood of each phenotype. So for a dominant cross, the phenotypic ratio would be 3 affected and 1 normal. The phenotypic ratio is the likelihood of each phenotype. So for a dominant cross, if this was Huntington's, the phenotypic ratio would be 3 affected and 1 normal. If the cross was recessive, so if we were talking about cystic fibrosis, the phenotypic ratio would be 3 normal and 1 affected. Or if you wanted to keep track of the carriers too, which is not the official way to do things, but it does seem important to keep track of the carriers, you'd say 1 normal, 2 carriers, 1 affected. Just to be clear, this is not a Punnett square. What I'm trying to show here with this table is that there's 3 possible allele combinations for each parent. So if we were talking about eye color, the 3 possible allele combinations are this. For mom, the 3 possible allele combinations for dad are here. If you add those up, there's 9 possible crosses. Also, since 3 of these crosses, if these are not X-linked, it doesn't matter where the allele came from, then 3 of these crosses are redundant. So for example, this is the same cross as this cross, or this is the same cross as this cross. So there's a redundancy in these 3. So really there's only 6 crosses. So the ones that are not in orange are the 6 crosses that you should use to create Punnett squares and calculate the phenotypic and genotypic ratio. So what I'm saying is for each of the mutations that are autosomal, whether it's dominant or recessive, there's only 6 crosses to work through. So you might want to work out all the Punnett squares for those 6 crosses. In making the infographic, and it's going to be available on the YouTube later, I wanted to be clear that the letters refer to alleles on genes. So that's why I put a letter or an allele on a gene. So these are the 6 possible crosses when now I say we're actually talking about genes with alleles on them. So it's the same thing as this, but I'm just using a little bit different figure to point out that we're talking about genes. I'd recommend that you try to work out the crosses first and then check your answers with the next slide or with the infographic that's available on YouTube. So the answers, the genotypic ratios and phenotypic ratios for these 6 crosses are on the infographic that you can get on YouTube. Or they're here too. So for the genotypic ratio here, you got 4 big B. If this was Huntington's, you'd have 4 affected. If this was Huntington's, you'd have the ratio of 2 big B, 2 little b. These 2 would be affected, and these 2 would be normal. So work out the Punnett squares and then use these figures to check your answers. For recessive genetics, the crosses and the genotypic ratios are the same. So this is the same exact table, and in fact, this is the same exact figure, but I would recommend that you work these out because the phenotypic ratios are now going to be different. So you see now, the one I pointed out before was this one. So now the genotypic ratio is still 2 big B, 2 little b's. But now we've got 2 carriers or 2 affected. Or maybe let's look at this one. If we go all the way back, we can see here that the phenotypic ratio was 3 affected and 1 normal. And now our phenotypic ratio is 1 normal, 2 carriers, 1 affected, or technically 3 normal, 1 affected. So we went from 3 affected, 1 normal, to now 3 normal and 1 affected. There's still just 6 crosses for X-linked, Things look different though, since we have an X and a Y chromosome. So across the top is mom, who has two X chromosomes. Along here are dad, who has a Y chromosome. Still 6 possible combinations of allele passed on by mothers, but only 2 possible combinations for the dad, so we get 6 crosses. Again, I would work through these, make your own Punnett square, make out the genotypic ratio and the phenotypic ratio, and then you can check your answer with the infographic or with this slide here. Pause the video and check. So those are crosses, and here are the videos if you wanted to watch the videos. And that's genetics. Thank you. Hey, that's the end of AP2. Thank you.